welcome back to the course on cell culture technology. Today we are into the third class of the first week. So, in the first two class, I made an humble attempt to kind of request you to think beyond cell culture as just a culture technique. And I told you that at every point of it, there is a philosophy, there is a philosophizing in the subject instead of blindly following certain things. And there is simple chemistry, physics and biology involved in it. So, today, the next two classes, 2 to 3, whatever, whatever it takes us, we'll talk about the biology of the cells vis-a-vis the in vivo and the in vitro conditions. Because once we understand the biology of the cells, it will help us to recreate a situation which is similar to that of inside the body. Okay? So, we are into week 1 and today is our class 3. Okay. So, today we will talk about the biology of the cells to be taken into account to grow the cells outside the body. Okay. So, this is the key point what we will be dealing next few classes. Okay. So, in order to address this point, first of all we have to understand under what conditions the cells grow inside the body. Okay. So, again let us take the example of one of the previous examples where you dealt with. Okay. So, say for example, if we take the same example again, and let us take the example of the cardiac or the heart. Okay. So, these cells which are growing out here, the cardiac myocytes, which I have already explained earlier, and here are the gap junctions. Okay. These cells out here have the first parameter, they need oxygen and they give out carbon dioxide and they need oxygen. So, there is something called, they have a mechanism of gaseous exchange inside the system. Very, very first thing you needed, because in the absence of oxygen, in its absence, which is an anaerobic situation, these cells will die. Okay. So, they need sufficient oxygen, and not only that, this carbon dioxide which is produced here has to be sent outside the system. Otherwise, this will make the environment acidic, because CO2 plus H2O, we know that it will form carbonic acid. Okay? So, that is something one has to keep in mind. So, there has to be a proper gaseous exchange which should take place. Without that gaseous exchange, these cells are not going to grow. So, first thing what one has to keep in mind, if you are taking out this tissue out here, and you should be able to provide the right milieu of gaseous exchange. Okay? And we will come to that, how the gaseous exchange, how this milieu is being maintained in a system outside the system. Okay? Parameter 1. Next parameter is this. We have to figure out, if you remember that in one of the lectures I give a pause, I say if these cells adhere to the surface. So, there are two kinds of cells. Cells which are not here, of course, there are other parts of the body they have. They are adhering cells and non-adhering cells. Which cells are we culturing? So, talking about non-adhering cells. Adhering cells, you could see, these are all adhered to each other, right, in our hand. Now, talking about the non-adhering cells, blood cells, they are circulating in your body. They are non-adhering, right? They do not adhere. If they adhere, then will collapse, right, because they have to carry oxygen. So, are we talking about RBCs, red blood cells, or are we talking about WBCs, white blood cells, or are we talking about, of course, RBCs means all the lymphocytes, leukocytes, macrophages? Are we trying to culture these? And if you are trying to culture them, do you want them to adhere, or we want them to, in a suspension to move around? What are we culturing? This is a very critical point. So, think of it, the hard tissue which is growing out here, it is exposed to blood, and that blood contains lot of non-adhering cells moving through. And those non-adhering cells, while they are moving through, say for example, this is a non-adhering cell. One second, let me change the color. Non-adhering cells, it is rolling, it is moving through. But while it is moving through, it may secrete out certain things, right? In a real life, we do not know what it is secreting, and that what it is secreting to its surrounding may influences the another cell which is an adhering cell sitting out underneath. If these pink colors are the adhering cells which are sitting underneath, and top of that this red cells is moving through by secreting certain XYZ compound, XYZ chemical, and this is influencing some of its biology. We really do not know that. So, first we have to decide, are we culturing adhering or non-adhering? At this point, I will stick to the adhering cells. We talk about the adhering. We are not taking into account the non-adhering cell. That's why I give a pause in somewhere in my first or second lecture that, are we talking about what I did not introduce, that complexity. So, here I thought that I bring it back. So, if we are talking about adhering cells, so the cells, the way it works is something like this. Say for example, I have a cell like this, okay. Here I have a nucleus with a DNA sitting there. You have all sorts of organelle, mitochondria, endoplasmic reticulum, ER, nucleus, MT stand for mitochondria, likewise you have the plasma membrane, PM, These cells have lot of membrane proteins which are sitting there. Now, if these cells have to adhere to a substrate or adhere to each other, they secrete specific cementing materials, and by virtue of which these blue arrows coming out are telling you about the cementing materials. These cementing materials helps it to adhere to another cell in its vicinity of its type or another type or whatever. GA is the Golgi apparatus likewise. Okay. So again, these two cells then form the colony. So say for example, these two secrete another set of cells and it invites the third one to form. So they are slowly forming a colony. So in order for them to adhere, these compounds which are shown in blue out here, this forms a matrix. So if you could remove all the cells, what you will be seeing behind, they will be left behind will be something like this. Those blue color arrows what I am trying to show. And this matrix is called, in technical terms it is called extracellular, because it is extra, it is not inside the cell, right? It is an extra, outside the cell, extra. This is not part of the cell. Extracellular matrix. It forms a matrix like this. Here is a matrix. And this extracellular matrix are mostly proteins and part of carbohydrates. And there are metal ions and several things which are involved in it. This extracellular matrix is one critical feature in our body which ensures the cells remain adhered to their specific locations, because this particular aspect is very critical when we will be talking later about cancerous cells. Okay? So this extracellular matrix is a kind of, you can call it, the signature of the colony of cells. What do I mean by the colony of cells? Now let me come back to the previous diagram. So this is the, I name it as the heart colony. Okay. Now out here where I have the brain, this is the brain colony. Okay. Now I have this stomach and everything. This is the, say, stomach colony. Then the series of things. So each one of them are colonies, and within colonies there are subcolonies. How is it so? Say for example, if I break open the heart, within it, say, the structure of the heart is something like this, where it has 4 chambers like this, Now within it, these are the 2 upper chambers, depending on which angle you are looking at it, from which side. L stand for left, R stand for right, UC stand for upper chamber, LC stand for lower chamber. So within this colony, there are multiple colonies, and each one of these, they have different cell types which are involved in it. Ventricular cells, those are the lower ones, are the ventricular cells, which are covering this part, ventricles. The lower 2 chambers are called ventricles, these are called auricles, and they have different cell types with mild variations. So they are different colonies, and different colonies leads to different extracellular matrix, the term which I introduced in the, so in short this is called, in biological jargon they call it ECM, extracellular matrix. is nothing but identification of that location. These cells grows here under these conditions. So, it is a very classic signature of extracellular matrix. Now, if I have to, coming back here, if I have to grow these cells outside, so you take out these cells, the first thing what I have to mimic is, so if I have these cells out here, I should have the right set of ECM, extracellular matrix. So, if I have to grow these cells outside on a dish, then it's the prime prerequisite is, if I have a dish like this, the dish should have the right set of extracellular matrix. So, I should be able to code this. I am just putting blue because since I showed you the extracellular matrix in blue, I am just coating it with blue. Should have the right set of extracellular matrix protein out here. Now, one can, since I am talking about extracellular matrix protein, so if one has a hypothesis saying that the, say for example, I mark this extracellular matrix as, say, I call this as blue. This blue is showing the extracellular matrix, okay, and I call it as B. Now, if I give you a hypothetical situation, if I say, if in the extracellular matrix B, you add compound P, which I am representing by, say, compound P, something like this. And if you grow these cardiac myocytes out here on top of this, then it will change the properties of cardiac myocyte. So, they will not adhere to each other. So, in that situation, you will only see the cells sitting there, they will not move. They will not migrate and form the structure what I showed you. Then if they have to form, they should show something like this. Like if you remember when I showed you this structure, they should show the electromechanical activity and all these things, they should come close to each other. Remember this structure which I showed you in probably in the first or second lecture. So, they will not be coming close to each other. Now, here I am telling you that you can actually test such things if you say this P will actually prevent their migration. And say for example, P is some form of a toxin which binds to extracellular matrix and prevent the cardiac myocyte to come close to each other. So, how to test it? Here is an example. So, you see, if you know the basics, then you can answer infinite number of questions, but you have to understand the basics very right, because as I will go through, you will see, you just culture the dish, and many people, as a matter of fact, because of lot of budding graduate students, they don't even understand the significance. They buy coated dishes to culture things without even realizing that there is a tremendous chemistry and biology involved in it. It is not something like just mix something, mix match something, something is going to happen. It doesn't work like that. There is a logic. And one has to, because it's a very tough process. I mean, one has to go through that whole grill to understand that what is the significance of having these kind of understanding. Or say for example, we start predicting that okay, these are ECM which are natural. These are natural ECM which consist of, as I have mentioned, of proteins and carbohydrates. Now, I have a synthetic molecule, a synthetic analog, which exactly mimics a natural analog. Big discovery, and we will talk about it. There are synthetic analogs which exactly behave the same way as their biological counterpart. How to discover it if you do not know the basics? And within scientific, within this, one can have synthetic things which may be an inorganic counterpart, maybe an organic molecule. But in order to decipher it, one has to understand the biology and chemistry behind it. Because I told you there is another very interesting thing which is related to this extracellular matrix protein. That is, coming back to this picture. Now, when you talk about carcinomas or cancer cells, so what are really cancer cells are? Cancer cells are originating from your own body. These cells, say for example, we talk about a colony. Okay, let me draw it something. Say for example, you consider your body as colony. It's a, say a blue colony, a red colony, a green colony, a dark green colony, a orange colony, a pink colony, a fade blue colony. Okay, this is good enough. Okay, and each one of these colonies have different, cells which governs them. Right? These are the different cells, and this whole system lives in harmony without, creating ruckus. Okay. 1 minute. Okay. There you are. Right. And they are connected with each other with blood vessels, and they crosstalk with each other. It's a system which is functioning in complete harmony with itself, and they are happy-go-lucky family. Now, say for example, in this colony, say for example, one of the kid become rogue. This is that rogue kid, and this rogue kid decided that I am going to create problem. What this will do, it will travel, it will come out of it. So now think of it. These colonies, identification of these colonies are their ECM. There is a different ECM here, extracellular matrix. There is a different ECM here. There is a different ECM here. ECM is their barcode. You remember the barcode? Whenever you buy some material, you see the barcode. It is the barcode of that cell which decides which part of the body it is going to grow. But here this rogue fellow decided that it will deceive the barcode, and it will go anywhere and it is going to grow. It will invade, it may invade this part, and it will grow like this because it is now more of a suspended cell. It can grow anywhere and sit. It does not have anymore the barcode to grow. It has kicked out its barcode. It goes here and it creates trouble. It may go here and it create trouble. Wherever it decides. It's a rogue fellow. So all of a sudden, a harmonious system goes for a different swing. And that's precisely what happens in a cancer cell. A cell which decides that, hey, I do not want to become part of this colony. How I cannot become part of the colony? I shut off my barcoding and I cheat this colony, the red colony. If I start with the red colony, I cheat the red colony people. I moved out from that colony. I went to the dark green colony or the light green colony or to the pink colony, and I spread myself. Because I do not have any barcode now, I can grow anywhere and as much as I can grow. This is precisely what happens, and that's why I am telling you, understanding this part of the biology, of understanding this extracellular matrix, again I am reiterating this point because try to understand the science behind it. Extracellular matrix is so very important. So please, as the next generation upcoming scientist, do not follow things blindly. Please think. There is always a chemistry behind it. Without that, biology doesn't exist. There has to be a basic fundamental chemistry behind it. So please think over it. I have seen people buying dishes coated with some XYZ extracellular matrix and they grow cells. Yeah, surely something will grow. I mean, who is stopping them from growing? Life expresses itself. There's a thumb rule. But then that's not the way to do the science. One has to understand the basic concepts. What is happening? If I change this, so it should be able to play with the cells. You know, if I change this extracellular matrix, this is what is going to happen. If I use this, this is what is going to happen. How the cell behavior changes. As a matter of fact, within the same extracellular matrix with slight difference, their physical parameter changes. So this is that part where when a cell becomes rogue, when the cell becomes cancerous, because this extracellular matrix signature or the barcode is compromised. So for those who are from outside biology, you can call it as the compromised ECM barcode. This is how you should remember it. The barcode has been compromised. When the barcode is compromised, they can cheat anybody, and they do cheat. They go to some other part of the body and then create ruckus. So this is how it works. So this is the second aspect what one has to understand, that what the ECM does. In the next class, we'll follow further with the biology of these cells, which will help you, because based on this, so we have talked about today about the gaseous exchange and how we mimic it. We haven't talked about the mimicking, but how we have to mimic that condition. Second, we talked about the ECM, right? We'll follow it up in the next class. Thank you, and thanks for your patience.